All right, so this is going to be the first of 3 chapters that we're going to cover, the last of your chapters, in a unit that we're going to call cardiorespiratory system. We're going to cover blood. We are briefly going to cover the heart and the circulation of blood through the body, very briefly, and then we're going to cover lungs and the respiratory system. So, blood is our first chapter in this unit. And I'm going to break this down just for ease of uploading into a couple different lectures. But I'd like you to submit all your notes as one. So it's going to be one assignment with several different lectures. All right, so let's start talking about blood. So I'd just like to remind you, way back when the world was different in August, we talked about blood being a connective tissue. And the reason that it is classified as a connective tissue is that it has cells. The cells in blood are white blood cells, red blood cells, and platelets. There is ground substance, which is the plasma that those cells float around in, and it has fibers. The fibers are dissolved in the plasma until you have a cut and they become solid, and that's what the coagulation process is about. So because it has all those components and it originates from mesenchyme, it is connective tissue. So remembering back to that. All right, so there's a test that you can run. It's one of the oldest and most basic blood tests that's called a hematocrit, where you take whole blood, that just like the blood that comes out of a person with a standard needle, and you spin it down in a centrifuge, and what happens is that the red blood cells go to the outside, the white blood cells and platelets go to the middle, and the plasma, the liquid component of the blood, stays on the outside because to the inside because it is least dense. So they go in order of density. And there is a ratio that you should have. Generally speaking, you should have about 45% red blood cells. That if you are very physically fit, you might have some more of those. But within normal parameters, you should not have less than 45% red blood cells. That would be a type of anemia. You should have about 55% plasma and about 1% or less than 1% of leukocytes, which are those white blood cells. If you have more than 1%, if you have a lot of white blood cells, that would be an indication of a kind of infection. And we'll talk much more about that in a future lecture. So, remembering that when we start these things, there's always a question that says, what are the functions of something? So the functions of blood, blood has multiple functions, and then I'm going to give you some examples. So the first function is distribution. So what does it distribute? It distributes, first of all, oxygen and nutrients. So the nutrients come from your digestive system and your liver, and the oxygen comes from your lungs, and they need to go to the body cells. If the body cells don't get that oxygen or those nutrients, they die. It also takes waste from the lungs, waste from those cells, and so the waste that comes out of the cells, so oxygen goes in, nutrients go in. Nutrients, let's just say glucose for now. What comes out of that is carbon dioxide as a result of cellular respiration and waste products of metabolism, and those will go to the lungs for the carbon dioxide to be exhaled, and we'll talk about that more in the next chapter. And then the kidneys, are going to take all of your blood in, every 2 minutes or so and filter out the bad stuff. In addition to that, we talked about the autonomic nervous system, and we talked about things like epinephrine and norepinephrine that get, and we talked about, more along the lines, less of that, and more of adrenaline, epinephrine, norepinephrine, getting dumped into the bloodstream and being transported, but there are lots of other hormones that are transported through the bloodstream as well. Sex hormones, growth hormone are all components that end up in blood. So the second function of blood is a regulatory function. So maintaining body temperature. We've talked a great deal about vasodilation under the skin, making your body cool, and vasoconstriction, pulling heat back into your body. So we talked about that a lot. Also maintaining a normal body pH, so keeping acids and bases under control. Your blood has buffers in it that absorb hydrogen and hydroxide ions that keep the pH around 7. And then adequate fluid volume. So you have fluid in your blood, water component, that can go into tissues if you need it. And that is done in accordance with your kidneys. And one of the things that happens when people become very dehydrated is that their blood becomes very viscous because you've taken all the water out of the blood that you could. There's a protective function of blood. Blood will coagulate if it needs to. So if you get a cut or one of your blood vessels tears, which is what bruising is, it's just bleeding where you don't tear your skin, you just tear the blood vessels underneath. There is a coagulation or a clotting pathway that keeps your blood from exsanguinating, from your blood to come out. And then the second part of this is that you have leukocytes, which is the name for white blood cells, antibodies, and other substances in your blood that make up your immune system. So a large component of your immune system resides within your blood in the form of white blood cells, and the rest of it would be in your bone marrow and in sort of your skin and your eyes. It's sort of, but a lot of the specific kinds of immune system is very much in your blood. All right, so let's talk about these parts of the blood again a little bit more specifically. So plasma is the liquid part of blood that includes proteins. Plasma would be something, and so if you work in a lab, which some of you will probably do someday, there are tests that go into machines, and sometimes you need whole blood, which would be what you see over here. This is whole blood. This whole blood would be something that sometimes you would run a test on. Oftentimes a test would ask for plasma, and plasma would be the blood that you have, whole blood, that you took from an individual, and inside the vacuum tube that you put it into was some sort of anticoagulant. It might be heparin, it might be a citric acid product, but heparin is the most common one, and it causes this blood to not coagulate and clot. So it looks like whole blood the whole time. You'll notice that when a phlebotomist draws your blood, he or she will invert the tubes gently. That's to mix up. Some of the tubes have anticoagulant in them, and they will cause that blood not to clot. Otherwise, it will clot within a matter of moments on its own, being exposed to air. So then you spin it, like I said before, in a centrifuge, and you have red blood cells, white blood cells, and platelets. And then this part is called plasma because it still has the clotting proteins present. Plasma, because it has clotting proteins, is used in transfusions for people who need clotting proteins, who have clotting disorders, the best known of which is hemophilia, which you probably learned about in the genetics component of your biology class. So plasma has glucose, which your body uses for energy, amino acids to make proteins, vitamins, which are generally speaking cofactors in reactions, hormones, mineral salts, antibodies, which are part of your immune system, antitoxins, particularly if you've been exposed to something that you have overcome. Antibodies being like antitoxin is the same thing as an antibody, but just for a different type of ailment, something that you've consumed that is toxic to you. Urea, which is a nitrogenous metabolism waste. phospholipids and lipoproteins to make cell membranes, hydrogen carbonate atoms, these are going to be buffers, and then fibrinogen, which is the liquid part, the liquid protein fibers that will clot when it's time for your blood when your blood needs to clot. So as opposed to that, you might have serum. And serum is actually used just as often, if not more often, because sometimes those protein fibers get in the way of testing for other proteins. So you want to eliminate them from your testing. So you take whole blood and you wait for it to clot. It doesn't take very long. You just leave it in the tube for a bit. And then you have 2 layers of that and you spin it down. And the serum is at the top and the blood is at the bottom, and the blood is like an actual like gooby blood clot. So it is used in testing and you just pull off the serum and use that serum in the testing, and it would have all of the things that we just talked about. Let's go back, but it wouldn't have that fibrinogen, and that might interfere with some of your testing. So that would be why that would be useful. All right, let's talk about some blood cells. The most numerous of your blood cells are erythrocytes, which are also known as red blood cells. Throughout this whole lecture, we are going to abbreviate that RBCs, and that's the way most people who write red blood cells or erythrocytes abbreviate them, RBCs. They look just like this. This is a scanning electron micrograph of a bunch of red blood cells. They look like discs or donuts that the hole is not quite gone through. They do not have a nucleus. They are born with a nucleus in the bone marrow, and then they become denucleated as they mature. And because they don't have a nucleus, they can't repair themselves, and they have a finite lifespan. So in the course of about 120 days, the erythrocytes that you have now will be dead in 120 days and replaced by a whole new set, presuming that everything is going well in your bone marrow. The process of making red blood cells is called erythropoiesis. Erythropoiesis is something that will definitely be on your test, and we're going to talk about it in a couple different contexts, but it happens, you make more red blood cells. Generally speaking, you have sort of a maintenance level of erythropoietin, which is the hormone that your kidneys make, and they make a certain amount of it, but you can make your kidneys make more. And you can make your kidneys make more by hypoxia, which is having low blood oxygen. You can get low blood oxygen by exercising. And this is like the hallmark, the baseline of being in shape. So if you are in shape, you are exercising to the point where at some point your blood oxygen dips because you're using all the oxygen that your red blood cells have. And then you would be, that's when you're engaging in anaerobic exercise. So your body starts doing anaerobic respiration. That's where you get like jelly legs. And so when that happens, your kidneys make more erythropoietin. When that happens, here's your stimulus. And the stimulus, so we'll talk about a couple other ones, but just say you work out, that you have an imbalance, so you don't have enough blood oxygen. Hypoxia is the word for that. And it reduces your blood oxygen levels, and it causes your kidneys to make erythropoietin. That travels through the blood to the bone marrow, and in the bone marrow, it causes more red blood cells to be made. That's called erythropoiesis. That causes your ability to make your oxygen carrying capacity, so the ability of your body to carry oxygen, which is the thing that makes your muscles work better for a longer period of time, to increase. So where you hear about this most is in athletics, and mostly the sort of cheating kind of athletics. There are some really great books about this. I can't remember them off the top of my head. They're mostly about cycling, but there are a whole bunch of different sports that, particularly endurance sports, track and field, and track and field, like long distance running, and particularly cycling. Cycling is best known for that, for blood doping, because it is such an endurance sport. So it turns out that things that are legal to do in sports are things that are training type things. So in order to increase your red blood cells, you can exercise really hard. Assume that if you're like a Tour de France level or Olympic level cyclist, you are already doing that. You are training as much as you can. You can also sleep in a hyperbaric chamber that decreases the amount of oxygen that you have the whole night, and that makes you even more hypoxic than you would be just exercising. You might have it maxed out, and that will biologically max out the amount of oxygen you can make. You can also go and train in a place that is way above sea level where the oxygen is thinner. That happens. And when that happens, it will also make you hypoxic. If you've ever gone and tried to just be a person in, Telluride or in the mountains of Colorado, you will feel like you are out of breath all the time. And it's actually really hard for people to adjust to that. And it takes a couple weeks for your red blood cells to kick in. So those things are all legal. The things that are illegal to do are things like inject yourself with erythropoietin, which on doctors or people, the person injecting you with it will call it EPO, because that's its doctor name. So you would get injected with EPO, so it bypasses this part and the working hard part. I mean, you're working hard. Let's just give that a given. And then you make more red blood cells. The other thing that's illegal to do is to withdraw your blood and freeze it while you are training and before your competition, and then as you're competing, inject your red blood cells into yourself. That is also not legal. So there's a lot of different. If this is interesting to you, I will throw up some links in this lecture series so you can check that out, because it is fascinating. I will also try to link to a movie about a guy who does it for a documentary, who blood dopes. It is really crazy. So worth seeing. Maybe we'll do that. All right. So moving on. This will definitely be on your test. So I would know hypoxia as the thing that is stimulated, but hypoxia is Hypoxia is stimulated by decreased availability of oxygen. That's like going to Colorado or training someplace in high elevations. Decreased actual red blood cell count. That's the top one, which means to say blood loss. So if you have a bleeding cut or some sort of anemia, that would cause hypoxia. Or increased tissue demands, that's exercising. So those three things would be the stimulant that causes hypoxia. Hypoxia is the reduced oxygen level in blood. That causes your kidneys to release erythropoietin. Erythropoietin goes to your bone marrow, in the red bone marrow, not the yellow bone marrow, and causes more red blood cells to be created. And that increases your oxygen carrying capacity in your blood and makes you able to exercise harder and longer. Just saying that this process, if you haven't left the house in a while and you haven't exercised, it takes about, it probably takes 3 or 4 days for this process to start tipping down. So you start losing your sort of in shapeness by not exercising. If you don't exercise for a week, when you come back on that week 2, you will know it. And one of the reasons is that you have fewer red blood cells than before. All right. So what are red blood cells made out of? They are made out of iron. Iron is a very important component of it. If you are iron deficient, you will not have as many red blood cells as you should. These are all things that can cause you to be anemic. So the iron is a component of your diet. Also, B vitamins are a component of your diet, and if you don't have either of them, you will not make enough red blood cells, and those red blood cells will not carry enough oxygen, and so you will feel like weak, tired, lethargic, all of those things. And then hemoglobin is the protein component that your body makes, and it incorporates both those B vitamins and iron. This is a great diagram because it shows you that the red blood cell itself, it's very dense, and it's made out of these hemoglobin molecules, which have 4 little iron molecules. The blue dots are on top are the oxygen that go to the heme part, and the heme part, it has the iron. That's what the heme means. So these hemoglobins are 4 units, protein units, that each hold 1 oxygen, and they're all folded up together and sort of chained up and make that donut-shaped, or sort of like the inner tube that you would sit in at the like raft like structure. And so that's all just hemoglobin, so it's all there just to carry oxygen. All right, leukocytes are the other solid components of blood. So if you look at this, this has all of them. These little pink guys are the red blood cells, or erythrocytes, and they are clearly the predominant thing. You can see that they don't have a nucleus, and I know that this is weird. We call these big purple things white blood cells, and they are white. But when you look at them, they've been stained. The stain that you use for this is called a Wright's stain. You can see here, Wright stain. And that is the very typical stain that you use to identify white blood cells. And the white blood cells pick up the Wright stain in a very specific way, and it makes them specifically colored. And you will learn all of the white blood cells in a future lecture. So generally speaking, all of these white blood cells have a specific immune function, and you have about 11,000 of them per milliliter of blood if you're healthy, and if you have an infection, you will get more white blood cells, and you will get more of a specific kind of white blood cell, which is why blood tests are often, if you don't feel well, one of the things your doctor will probably do is blood work. And one of the things that they will do in that blood work is to count your white blood cells, and look at how many specific ones there are. And so the purpose of each one is specific to what it does. All right, so we will pick up here next time.